I hope you had a lovely break. So what we're doing today is we are going to continue on through chapter 12, and we'll be talking about neuron structure and neuron function. So that's our goal for the day. Friday, of course, then is the joints and muscles mini exam. I think no, unfortunately, we cannot. So that is when we do need to do it. I don't know. I was answering a couple questions earlier, but do you have does the class have questions or other people have questions about it? Yeah.
Are you gonna try to trick us?
No. My intention. Okay. Yeah. So my intention, and there's always intent, and then there's how I evaluate what ended up on the paper, and then how you evaluate what ends up on the paper. But my intention is that there shouldn't be anything surprising on there. There shouldn't be anything that is even, hopefully nothing that's even ambiguous, in terms of what would be the proper answer. So one thing people are asking, and I might have said this before, but I don't remember, and somebody's asking, so I'll do it again, is structure of questions. For the most part, like my intention is, again, it's intention, but I haven't written the thing. That's on my to-do list to hopefully get done today, but I don't know when that's going to happen. Is I will, like for the action section, I will say, which of the following muscles does this action? And I'll describe an action as stated in the blue text from the Merib textbook that I posted online. And then you will pick the correct muscle from the list of optional muscles that are down there. And I will try not to have a situation where there are multiple muscles that do the exact same action. There could be a case where you have a muscle, like I might list an action that does like a couple different things. So maybe there's a couple different actions a muscle is importantly involved in, and they're all listed in blue text. And there could be one muscle down there that does like only one of those, but it doesn't do all of them. So again, I'll try to make it clear and not, where there's clearly a correct answer. Yeah.
For like the action, will you do it like you give us the action and put two muscles, one that is a prime mover and one that's just like a secondary?
Not unless the action states what is the prime mover of this action. Yeah. So, so an example, so if the action is closes the jaw, elevates the mandible, I would not put masseter and temporalis unless it said what is the prime mover of jaw closure, and then I would put masseter. But otherwise, only one of those would be listed as an option. Again, my intention is to make it not ambiguous as far as what the correct answer is. The same would be true for attachments. So I will give you an origin, or I will give you an insertion. It'll say, which of the following muscles inserts at, and then I'll spit back text from the Merib textbook, and then you pick the right muscle for that.
So the choices of answers are going to be the names of the muscles?
Yeah, for those two parts. For the muscle actions and then for the origin insertions, the choices will be the names of muscles. Probably. I don't know. I'll try to put even subheaders in there. But yeah, we'll do like, all the origin insertions will be in one spot and all the actions will be in another spot. And you'll kind of know as you're transitioning through. Yes. So there's the actions, that's kind of like part 2 of the muscle homework. There's origin insertions. That's part 1 of the muscle homework. Because those are the only muscles you need to know attachments for. And then the last bit is the joints and muscles table, which, oops, I'm in the right thing. I just need to get there. So just to remind you, make it super, super crystal clear, I'm in the right chapter. So this is the answer key that I posted for the joints and muscles homework. And so I am not going to be asking things for that question or that question, but I will be asking questions from these tables. So you do need to know the locations and functions and such, and I'll probably use this language that's on my answer key. Okay? So it's, it's mostly, for the most part, I think for these guys, if you know where the thing is, you probably can figure out like what its function is, because they're usually sort of opposing a movement from that direction. But in any case, this will probably be, just like the Merib text, will be my basis for writing the other questions for about muscles, for the joints, or for the ligaments, tendons, bursae, menisci, what else is in there? Labri, I think it's be plural for labrum. I'll be using this thing. Okay? Other questions? Again, it is going to be multiple choice. That's not what I do in blood vessels in the spring, but for this one, I don't know, I feel like it's enough stuff to learn that multiple choice is appropriate. Other questions on that or anything else? And the great thing about it, I just saw you, go ahead. I don't think I can tell you right now. Like, I just don't know because I haven't started thinking about it. It will definitely be completable in the time allotted in the class. And that is the only thing we are doing on Friday also. So, you have the whole period. So I don't know, like it could be up to 50 total questions. Up to 50 total questions, which would be 1 minute per question, which means some people will be done in like 20 minutes because people just like, huh? Yeah, that's putting a lot of stakes on that one question, right? Be like, what's the hardest possible origin I could ask you? Yeah, no, we're not doing that. Other questions? I was about to say a thing. Oh, I know what it was. The great thing about this is that after Friday, I don't want you to so what I did at the end of organic chemistry, and I don't know how many of you guys are going to have to take organic chemistry, but at the end of organic chemistry, I literally went into the bathroom and I flushed the toilet to symbolize all the knowledge leaving my brain and going down the toilet, and it is literally gone. Like, I do not know what an ester is, and I probably shouldn't tell you that, but I don't know anymore, and I'd have to look it up. So, I don't want you to purge all knowledge of this afterward because you will need it in future classes. I promise you. You're going to need to know some of these things, and many of these things probably. But it's not going to reappear in a meaningful way on the exam, except for the one thing I told you is that innervations will be appearing on the final. We'll talk about innervations, but they are very compartmentalized. So all of the muscles in the sort of front of your leg up here are all femoral nerve, and everything in the back of your leg is sciatic nerve. So it's pretty easy to do the innervations if you think about it compartmentally. Not for this. They're on there, but you don't need to know anything. They're kind of there as a reference for later. But you do not need to know any nerves for this exam on Friday. Okay? So again, most of it is kind of never again, except for after. Yeah, go ahead.
So you said that when you're going to explain the functions of a muscle, it would basically just be what's in the textbook. So you're not going to put in muscle action power to dunk the basketball. What muscles are these type of deals? It's all just going to be the textbook.
Probably not, unless I feel like I could just add a small little additional thing that would be clarification. So there was a question on I think it was on the quiz where I might have said something about the upward swing on a jumping jack. And that was in addition to some other statement of the action that was more anatomical and probably based on book. So I guess I might do that if I feel like there's some compelling reason for me to provide additional explanation. It will not be in an intent to confuse. It will be an intent to clarify. I'm sorry. You guys, I know this is really stressful. I'm trying to make this as straightforward as possible. That is my goal. I will do my best. All right. All right. The other thing I wanted to say is once that is done, so we'll kind of, that is the only thing I know that for this class that you'll be thinking about until between now and then. Once that is done, I still do want to do a quiz on chapter 11, which is the muscle tissue chapter. And probably that'll be due over the weekend, although I might let it bleed over into the next couple weeks, because again, or the next couple, maybe it'll be due Tuesday night or something like that. That might be better. But anyway, I'll try to get it up there. I just didn't get it written over break because I was doing other things. I don't know, other things. Administrative stuff. Okay, so here's where we are. Let's launch into this chapter. So we have already done a very brief intro to the nervous system. I feel like we didn't quite finish this. I'm going to go back to that slide in a second. We've pretty much done an intro to the nervous system, talked about subdivisions. And then what our goal for today is basically is to introduce neurons, neural regeneration. So there's kind of a basic structure function of neurons, and then start into this discussion of how neurons communicate. And that's most of the chapter, is that sequence. So it's kind of analogous to what we did with the muscle tissue chapter, where like a big chunk of that chapter was trying to understand how do skeletal muscle cells get excited to contract, and there was a whole bunch of steps. That's kind of what most of this chapter becomes, is how do we understand the function of a neuron in terms of their sort of resting state, how they get stimulated, how they fire action potentials, how they release neurotransmitters, and then what that does to the targeted cell that they're interacting with. So that's going to be our goal for the rest of this chapter, which is going to go well on into next week. I think we did end on this slide, and I was kind of running out of time, so I just did want to restate. So over in the peripheral nervous system, I know we talked pretty well about sensory division, and we talked about how there's somatic sensory division is basically all senses coming in from outside, whereas visceral sensory division is all senses that are from the inside of us. So somatic sensory would be eyesight and hearing, but also touch and temperature from your skin, that kind of input, whereas visceral sensory might be like your blood temperature and blood chemistry and whether there's food in your stomach and that kind of thing. We also have motor division that sort of mirrors that. So the somatic motor division is all the output that's kind of, you could think of it as kind of like being expressed onto the outside world. So it's basically all the motor output going to skeletal muscle is somatic motor division. Whereas all the motor output, we call it motor output, but the output from the nervous system going to our digestive glands and our sweat glands and what's something that's not a gland? I don't know. Blanking on some things, going to your digestive organs, informing them about whether they should be contractile or not, these kinds of things. Output to your blood vessels that's regulating their diameter. Those things are all visceral motor division, visceral motor output. And another word for visceral motor output or visceral motor division is the autonomic nervous system. And we have a whole chapter on the autonomic nervous system that we will do, I think, 3 chapters from now. Later in the semester. So we'll spend a lot of time on this. But the autonomic nervous system, which is the visceral motor division of the peripheral nervous system, has a sympathetic division and a parasympathetic division. Sympathetic division is your fight and flight system, so it gets kicked on when you're excited and energetic or scared or something like this. Whereas parasympathetic division is your resting and digesting system. So this is when you are tired and you're sitting around post-Thanksgiving meal digesting and falling asleep watching football, those kinds of things. That's when your parasympathetic division is running. So we will spend a lot more time on that later in the semester, but I did want to make sure that was clear because I think it got rushed. Okay, so what I want to do now is talk about neuron structure. And we did a little bit on neuron structure, I think, before, going back to the tissues chapter. So we talked about how neurons have this central structure. It's called a cell body, or the text is microscopic here, but in this case, it's labeled the soma, but that's the cell body. That's kind of like the cell proper for the neuron. That's where almost all the organelles would be, where all sort of normal function like protein synthesis stuff all happens in the cell body. And then you've got these dendrites, which are basically functioning as antennae. So these are picking up signals from other neurons or from the environment, depending on what kind of neuron it is. And then there's up to 1 axon. So this is the axon. They do show it kind of splitting, but there's basically 1 axon that then splits in multiple directions. And this is the output signal. So most neurons, I think I can say, I actually can't say most neurons. Most kinds of neurons have an axon. And these axons are conveying signals from this neuron out to some other cell, usually some other neuron, but it could be to a skeletal muscle cell, for example. Neurons are crazy big. So like one of the things that hopefully you see when you've did the tissues chapter, the tissues exercise in lab, and you're looking at slides of nervous tissue, the way you recognize that you're looking at nervous tissue is you find a neuron. And there's like a gazillion little tiny nuclei, which are all the little glial cells that are in those slides. But then you finally come upon a neuron, and it's like this giant gargantuan cell body that you see stuck in amongst all the other cells. So they're really big. And then on top of that, these axons can be absurd in terms of their size. So I got this sort of connection from some book. I can't remember if it was yours or not. But if you think of the soma, the cell body, as being roughly the size of a tennis ball, like if we scale up an axon, or you scale up a neuron, if the soma or cell body was the size of a tennis ball, the dendrites coming off that could potentially fill like a 30-seat classroom. That's this size of a classroom. So they could basically, you've got this huge antennal array coming out of this one little cell body. So this little cartoon illustration here vastly underestimates the typical range of how broadly these dendrites are going out. And then the axon, you could think of, it would be roughly the diameter of a garden hose, and you're sort of plugging it into a tennis ball. And then it would go something like a mile in length in some cases. So that's absurd, right? Like that's a garden hose literally going a mile out from this one little cell body. And yet that's the sort of reality that we're dealing with in terms of absurd sort of dimensions when you're talking about a neuron. So the result is that neurons are extremely reliant on, if nothing else, really sophisticated transport systems. All eukaryotic cells, all of our cells have transport systems because our cells tend to be fairly large compared to bacterial cells, where they are moving materials in and out, from the cell membrane down to the nucleus and back again. The neurons are no exception, and they have a really sophisticated network to do this, because if you think about it, if they're doing all their protein synthesis, for example, up here at the cell body, and then they're trying to send materials out to an axon that we just sort of reported is just an absurd distance from the, from a cellular level. These are very large structures. They need some sort of a transport network where they can not just rely on diffusion, but they need stuff to literally be pulled all the way from the cell body way down here to the axon terminals at the very end of the axon. We were doing this sort of metaphor, the tennis ball and the garden hose and stuff, but like there are literally sensory neurons that begin at the tip of your toe, and they have then axons that start there at the tip of your toe and then travel all the way up your leg, all the way into your spinal cord, and then the axon continues without ever, it hasn't talked to some other cell yet. It continues all the way up to the very base of your brain and your medulla. So, I'm, like 1.5 meters long. That's a single cellular process going that entire distance. So the only way you could ever have materials making it from the cell body out to the tips of those axons is through a transport system. You can't just rely on diffusion to make that happen at any kind of a rate that would be useful or appreciable for them. So they have this. Like all cells, they have these little motor proteins, which hopefully you've been paying attention to me and not this little thing that's kind of hypnotic. But what you've got here, this is a picture of a little motor protein. It is walking on a microtubule. So microtubules are one of those major cytoskeletal elements we talked about, and it does say that right there. So those microtubules, we have long axons. So if I draw a little neuron here, so that's my neuron. I'm not drawing the dendrites. Here's my axon. That's my axon terminal. I always draw it that way. I don't know why. But they've got microtubules that are running the full length of these axons. Sometimes they might break, and so you'll have little, multiple microtubules that are kind of running different lengths. But the idea is you've got microtubules running the full length of the axon, and then you've got these little motor proteins that literally do look like little feet that walk, and this is what they do. They are going to be grabbing onto vesicles, which it's a little bit hard to see in this image, but the vesicle is sort of tracing right along here. So it's this basically gigantic sac that it's pulling along, and it's bringing with it proteins. So these are little proteins embedded in that vesicle. There could be stuff inside the vesicle as well that are being transported from one end of the cell to the next. If you are transporting from the nucleus, technically it's from the centrioles. So if you're transporting from the cell body, basically, though, where the centrioles are, out to the ends of the axon, we call that anterograde transport. And anterograde transport is powered by a particular motor protein called kinesin. So kinesin is the specific little motor protein that walks along here. If you're going the opposite direction, and we do have transport that needs to go from the axon terminals all the way back up to the cell body, you're doing retrograde transport. Retro means sort of reverse. Like I have a retro toaster in my kitchen that looks like it's from the 60s, even though it was some cheap thing that we bought in the 2000s. But it still toasts bread adequately. But it's a retro toaster because it's going back in time. Here we're going retro, we're going back, retrograde. And in the case of retrograde transport from the axon terminal back up to the cell body, there's a different motor protein that kind of looks the same for more or less, but it goes in the opposite direction. It's called dynein. And these things are transporting materials around. I got a little video. I thought I had a little video. Hang on. Moving materials back and forth, because otherwise, basically, the ends of your axon terminals would just starve for resources. They won't have what they need. Which then creates a problem if you have damage. So one thing that can happen in the peripheral, well, it can happen anywhere in the nervous system, is these axons can be damaged. If it happens in the peripheral nervous system, there is some possibility for regrowth and regeneration. Basically, these in the peripheral nervous system, neurons can repair themselves. And so that's what's depicted here as far as how this process works. So here you've got a sort of a nice, healthy nerve fiber. So this is basically just a neuron, and they're showing its axon. And one thing that you can see is that there is, I didn't tell you this term yet, but there's this little layer of connective tissue, just like we saw with muscle tissue, that surrounds each individual neuron as it's passing through the peripheral nervous system, and that's called endoneurium. We'll come back to that term later on, but that's just like a little band of connective tissue that serves as a sheath around, within which that axon is traveling to its targeted destination. And the other thing you can see in here are these little tiny cells. These are glial cells. They're Schwann cells, which again we'll get to. I think that comes up on the homework actually. But their job is to basically help to nurture and maintain the axon, and they also dramatically speed up the propagation of signals down that axon. So what happens in this picture, though, is that you're starting with a nice, healthy, normal neuron, but then it gets damaged. So you accidentally cut yourself with a knife or something. And as a result, you've got this localized trauma where you have destroyed the axon in this area. It has been cut through. And the glial cells in this area have also been cut through. And so at this point, because the axon terminals, all the, down here, are reliant on materials coming from the cell body, getting down to them, they no longer are getting any of these materials, and so they all die. So the downstream end of that axon is going to die as a result of this damage, this cut that you've done by accident. So we've destroyed anterograde transport, and as a result, I said, why would this doom the axon? Stuff can't get down there. Protein products, for example, cannot get down from the cell body to the ends of the axon, so they're just going to starve and die. But what will happen is you'll get a response. There's sort of you'll see the nerve fiber will sort of start to swell. There'll be growth factors that will be released in response to this. I don't know why they swell, but they just do. But the key thing is that these damaged neurons, in response to getting these growth factors that are coming from neighboring cells that are sort of responding to the fact that there's been this damage that's occurred, they will start to sprout new growth processes. And that's what you're seeing down here, or at the end of what had been the severed axon, this cell starts to sprout little processes that kind of pop out here and there. I'm going to the next slide. And some of these processes will make their way into what's called a regeneration tube, which is where new Schwann cells, which are those glial cells, will regenerate down here. So they're dividing and propagating. They will get into the endoneurium tube, which is that connective tissue sheathing, and the result is you get almost like a little straw that leads your way down to where that axon is eventually supposed to go. And some of the growth processes that came out of the damaged end of the axon will start to make their way down this regeneration tube, and eventually, as they keep growing and growing and growing and growing, they will get down to the target cells that neuron originally was talking to. In this case, it's a couple skeletal muscle cells. There are special cell adhesion molecules that kind of guide these growth processes toward the targets. And ultimately, ideally, doesn't always work, but sometimes it does, ideally, you will get restoration of function, where these neurons are able to find their target cells again, they start talking to them again, and you restore normal growth or normal function with this regenerated neuron. That's ideal. Like my wife was washing dishes. Usually I wash dishes, and this is part of why I wash the dishes in the house, because she was washing dishes and she had this knife, and I don't know, she might have been mad at me about something that I did, which I was probably wrong about. But we had this stupid knife that had a cardboard sheath. Why we kept putting it in the cardboard sheath, I don't know, but she like slammed the knife into the cardboard sheath. Knife cut through the cardboard sheath, went into her finger, and she had a nice little gouge out of her finger. Took her to the ER, took a couple days to heal, stitches, all that jazz. But she never actually got full touch function back from that little swath of skin at the tip of her finger. I think it's actually her middle finger. So it doesn't always work, but in the peripheral nervous system, oftentimes there is potential for regrowth and regeneration like this, where you can restore function. I keep saying peripheral nervous system. In the central nervous system, it doesn't work, and it's frustrating as hell that it doesn't work, because this is the problem with spinal cord injuries, right? So if somebody has damage to their spinal cord, oftentimes there's so much scar tissue that forms, it's apparently a fairly regulated process. You have lots of glial cells that invade and cause the scar tissue to form. There are inflammatory signals that sort of prompt that scar tissue formation, and actually those inflammatory signals that are released at the site of the injury directly inhibit growth, which is one of these things where probably most of the time this kind of an inflammatory response inhibiting growth is probably a productive thing for cells, but not when you have an injury like this. And so the result is this kind of counterintuitive thing where you have signals that are released by your body that are directly inhibiting growth and inhibiting healing. So a major focus in spinal cord research is how can we turn off those signals, and especially in the early goings, right after a spinal cord injury, and promote regrowth and healing of those neurons in much the same way as already tends to happen in the nervous system. Because this seems to be the major reason why we can't get the central nervous system to regenerate. So that is a major line of research that you see a lot of papers on every year, where they're trying to work on that process. But anyway, the big point here is, how do neurons heal out in the peripheral nervous system? Any questions on that process? Okay. Regeneration tubes, glial cells, all that jazz. All right. So with that done, we know about neurons now. We know about major divisions in the nervous system. We know a little bit about how they can heal themselves when such an opportunity arises. What I want to spend the whole rest of the chapter on is trying to understand this basic question of how is it that neurons work? How do they communicate with one another? And so that requires us to understand kind of what neurons do in isolation, what happens when they get stimulated, and then ultimately how do they communicate with the next cell down the line. Because most of the time neurons are picking up signals from other neurons or from the environment, and then they're sending signals to some other cell, be it another neuron or some other cell within your body. So I have three major questions that we're going to try to answer as we work through this big picture question of how neurons communicate. The first one will be, how do neurons generate electrical signals? Because they are communicating electrically and sending electrical signals down their axons. So how does that happen? How do these messages get passed from one cell to another? Most of the time that's a neurotransmitter function, but not always. And then how does all of this allow for decisions, memories, thinking, etc.? I will tell you right now, we're going to briefly touch on this, and neuroscience, we don't know the full answer to this, but we have ideas. Like, there are testable hypotheses that are out there that help us to start to make sense of what a memory is and how decision making happens. And maybe a little bit on like what consciousness is, although that's I'm not sure I have a great handle on that either. So, how do we get into those sort of higher level cognition things, starting with a couple neurons talking to each other? And of course, the answer is, in part, that there's a whole lot more than just a couple neurons talking to each other. All right, so we're going to be working through these questions as we go through. The starting point to the first question, how do neurons generate electrical signals, is we have to go back to this idea of a resting memory potential, which is coming out of the last chapter, the muscle tissue chapter. And this is why I like to do these chapters sort of side by side, because neurons also have a resting memory potential. And when we're talking about electrical signals, we're ultimately talking about disruptions to that membrane, that memory potential, that resting memory potential. So to remind you how resting memory potentials work, you always start off with the assumption that there are some pumps somewhere, usually it's the sodium-potassium pump, that have established previously concentration gradients for ions. And in the case of neurons, just like the case of muscle cells, the most important neurons that are determining anything about memory potentials will be sodium, and especially for resting memory potentials, potassium ions. So these are potassium ions right here. And the memory potential is going to be determined by whatever ions are moving across the membrane the most. So whichever ion is the most permeable, and so therefore whichever ion has the greatest flux across the membrane, that's going to pretty much determine what your memory potential is at any given moment. In a resting neuron, it says resting cell, but we're talking resting neurons, but it is frankly true of almost every cell in our body, potassium ions are the most permeable ion. And the reason for that is that we have lots of these little potassium channels. These are, I call these potassium leak channels, because they are always open. So they just allow for a steady leak of potassium ions from the high concentration that they have inside cells. They're pumped in there at high concentration by the sodium-potassium pump. And they allow potassium ions to steadily diffuse out, which there they go. And as those potassium ions diffuse out of the membrane, they're going to create a little bit more positive charge on the outside of the membrane, which means that on the inside of the membrane, which is where we measure memory potentials, we have a bit of a negative charge. In neurons, it's usually somewhere around like minus 55 to minus 60 millivolts. I probably won't ever ask you that specifically, but it's you know, that's in the ballpark range that we saw with muscle cells as well. Muscle cells do this exact thing, right? So they have lots of potassium leak channels. They have sodium-potassium pumps that pump potassium in at high concentration, and then as the potassium leaks out of the potassium leak channels, it causes the outside of the membrane to get more positive, which means the inside of the membrane, which is where you measure the memory potential, is always a bit negative when they are at rest. The exact same thing is going on here. So you can flip back to those slides in that chapter and see it's basically all, everything here is basically being reported in the same way. So a resting memory potential, we measure that inside a cell. It's usually in millivolts. And again, common resting memory potentials are somewhere, I said, around like minus 55 to minus 60. Some of them will get down to like minus 75. Skeletal muscle cells will get down to minus 90 sometimes. But again, I probably won't ever ask you that, except that you got to know it's negative and you're measuring in millivolts, and it's not like negative 1,000 millivolts or something. So it's, you know, you should know the general range-ish, but just at least as in terms of reference, so you can then react to anything that's unusual. So I'm using some terms here. I'm talking about memory potentials. I'm talking about, I will be talking about currents, mentioned voltage. So I have a couple electrical definitions I want to give you just so we have some sort of grounding in vocabulary when we're talking about ions moving across membranes causing memory potentials. So there's really three major things that we're worried about when we're talking about electrical movement of electrically charged particles. One of these is current. So current is the flow of charge from one point to another. So that basically means that you've got positively or negatively charged things moving from one spot to another. That's a current. And you can measure currents in amps, or in the case of biological systems, it's always milliamps, which are, it's kind of like a millimeter instead of a meter. So we're dealing with fairly small currents compared to what you might have in your, you know, the electrical device in your pocket, in your phone, but they're still measurable movements of charge from one place to the next. So we could say that resting cells have an outward potassium current. That means that potassium ions are flowing out of the cell. That's all that means. Okay. We sometimes are also going to be interested in this term resistance. Resistance would be a hindrance or an opposition to charge flow. So this is usually measured in ohms, which is named after a person who had the last name Ohm, so I probably should capitalize it. So we might say, for example, when voltage-gated sodium channels open during an action potential, membrane resistance to sodium declines. Okay, so the resistance, again, that's the hindrance to the charge flow. There is another term that, I can't remember if I have it on here, so I'm going to write it up on the board. Yeah. There's another term that is related to this. It's called permeability. I just used that on the prior slide. Permeability is the inverse of resistance. Permeability. So I'm going to call it P because I can't remember if there's another abbreviation for it. But permeability is the inverse of resistance. So that means that if something here, if we say that the resistance to sodium declines, we would say at the same time the permeability is increasing. So it's, and that's because we're opening channels, right? If you open channels, allow sodium ions to start rushing across, we have decreased resistance and increased permeability to that ion, just by open up some pores that that ion can get through into the membrane. Final terminology has to do with this idea of a potential. So a potential difference in electrical terms, this is a charge difference between 2 points. In biological systems, we are almost always talking about potential differences across membranes. That's always what's relevant to us. So we might say that the potential difference across a membrane is called a membrane potential. So oftentimes we'll use this term, membrane potential. That literally just means, though, this charge difference between 2 points. So on the last slide we said we had more positive ions on the outside and more negative charges on the inside. So that, I'm going to go back. So here we've got a potential difference. That's a membrane potential across this membrane because there's more positive charges over here and more negative charges over here. So that's all that term means. The potential difference means there's some kind of charge difference between 2 points. Usually that would be the inside and the outside of a membrane. We measure potential differences in voltages, or in the case of biological systems, we're always measuring them in millivolts instead of volts. So, volts, like your AA battery is 1.5 volts. We are dealing with millivolts, so maybe 50 millivolts instead of 1.5 volts. But again, it's the same basic concept. It's just they are smaller voltages than you're dealing with when you're talking AA batteries. These are all related to each other using by this little equation. And I'm probably never going to ask you to type numbers or put numbers in there and do a calculation. But you should be able to sort of intuit your way through this equation and use this equation to just sort of think about what we're talking about here. So what this is basically showing is that your current will increase when you have an increase in voltage. And your current can also decrease when you have an increase in resistance, because they are inversely proportional to each other. So that should make sense, right? So like current and resistance. So this is saying that as your resistance goes up, maybe you close a bunch of channels, that's going to cause your current to go down. Right? So as you close sodium channels, you increase the resistance, which will mean the current of sodium ions goes down. Right? And conversely, if you decrease the resistance by opening a bunch of channels for sodium ions, that will increase the current of sodium ions that are leaving a cell. Actually, no, they would enter a cell. But anyway, that's the idea. So we're increasing the movement of molecules whenever you have a decrease in the resistance, and vice versa. The voltage is basically informing you how these are related to each other. So you could think of it as for potassium ions, if you have a really large current of potassium ions streaming out of a cell, that's going to result in a larger potential difference across that cell membrane, right? Because you got more ions leaving, so there'd be more positive charge outside, more negative charge inside, so the voltage reading would be larger whenever there's a larger current. So these guys are all related to each other. I'm kind of introducing this here. You should have a basic idea of how these things work. You should, again, I'm not going to ask you to plug numbers into it, but you should be able to sort of use that equation to sort of to understand relationships between current and resistance and potential difference. The other thing I'm going to preview for you is that we actually use this more when we start talking in cardiovascular system about how we regulate blood flow going through our cardiovascular system, because we can do what's called the Ohm's law analogy for some special word for it, but Ohm's law analogy basically for blood flow. And there, instead of current electrically, we're talking about flow of blood. Voltage, this can basically be the equivalent of pressure differences, and then resistance we still call resistance in that chapter. So we actually use this same sort of concept as sort of in an analogous form when we're trying to understand what drives blood flow around within your cardiovascular system. So you'll see it again next semester, is the point of all that. Okay, so given all that, I'm going to give you sort of a different kind of a scenario. So this question is asking, how do you think a membrane potential would change if a channel opened that allowed chloride ions to enter into a cell? So we've never dealt with chloride ions before, right? But here you've got chloride ions that are able to pass through a cell and enter into the cell. So what should that do to your membrane potential? And again, we're sort of assuming, we're starting from rest, but what should that do to your membrane potential, do you think? Chloride ions start streaming into a cell. Should it become more positive? Should it become more negative? Yeah. Does that make sense? So they're negatively charged ions. If they are entering into this cell, they'll make the inside of the membrane even more negative than it had been before. So you could have over here, you could have potassium leak channels allowing potassium ions to leave. They're positively charged ions leaving a cell. That will make the inside of the membrane more negative. But if you then open additional chloride channels, chloride ions could come in and make a membrane even more negative. So the point of this question, in a way, at least in part, is that what matters in terms of understanding how an ion is going to influence a membrane potential is which direction it's moving. So in this case, chloride ions are going in. And what is the charge of the ion? In this case, it's a negative charge. You kind of don't need to know anything else. Like, it doesn't really matter what kind of ion it is, except that we need to know its charge. So we need to know its direction of movement, and we need to know its charge, and that will determine how it influences a membrane potential. Any questions so far? All right. So at this point, hopefully we understand membrane potentials. We understand then how we could disrupt the resting membrane potential by opening additional channels and allowing new ions to start coming in or out. So sodium ions could start coming in, chloride ions could start coming in, and that will alter our original resting membrane potential that was established by potassium ions streaming out. So let's start working through and talking specifics about neurons instead of this more sort of general theoretical stuff, so we can better understand how neurons work. And we'll start with step one, which is that we're going to stimulate a neuron. And we're kind of going to do this now, and then we'll kind of come back to it again when we get to later on when we start talking about how neurons are communicating with each other. But we're just starting with this neuron. This is our focal neuron. And it is being stimulated somehow. Neurons are basically designed to collect disturbances and then convey those disturbances to other locations. So they're allowing themselves to be disturbed, and then they're conducting those disturbances in memory potential to another location elsewhere, often as it travels all the way down their axon. So initially, if you're looking at one particular neuron and you're trying to figure out when is it going to fire, it will usually be first disturbed by some kind of a stimulus that's impacting that particular neuron, usually somewhere out on a dendrite, although sometimes it could be on the cell body itself. And there's two basic kinds of inputs that they can receive. One type of input, probably the most common type of input, is when they're receiving a neurotransmitter, I'm sorry, I did this wrong. Let me start over. One kind of input that they can receive is when they're getting information from the outside, or from sort of some other cell or from some non-neuron, basically. So we call these receptor potentials. So this is basically what our sensory neurons are in the business of doing. Sensory neurons have some kind of a receptor that's sensitive to some sort of an input. And I know I'm being vague there, but that's because the input can be really vague, or can be really varied. So some sensory neurons are picking up light information. Those would be the ones in your retina. Others are picking up chemosensory information. So they could be detecting, like your smell receptors in your nose, could be detecting chemicals that are in the air that you just breathed in. Others in your skin are sensitive to pressure or to heat. And so each of these has a different kind of receptor, and those different receptors are sensitive to these different kinds of stimuli. But when they receive the receptor, the same thing kind of happens. Or when the receptor receives a stimulus, the same thing happens. So if a neuron within your eye receives light information, or if a pressure receptor in your skin receives pressure information, that will cause what's called a receptor potential, where there'll be some change in the membrane potential on the dendrite that just received that sensory information. That's sort of the first disturbance that this neuron ever receives. So you imagine that there's little disturbances out here, and that's the first, that receptor potential, that change in memory potential, when you bind your stimulus to your receptor, is what is the first sort of input that this neuron is ever going to receive. So we call it a receptor potential because it's the potential, it's the change in memory potential that occurs when this neuron receives its stimulus. You also have the more common scenario, which are postsynaptic potentials. And so postsynaptic potentials are those cases where you have a receptor that's receiving a neurotransmitter, and the neurotransmitter is pretty much always going to be coming from some other neuron. So this is probably the most common case, especially in your central nervous system, where basically all the cells are talking to each other. And so they are talking to each other primarily by releasing neurotransmitters, which then get bound onto receptors and cause a change in membrane potential. It's the same basic phenomenon here, right? So you bound your stimulus, and then it changes the membrane potential out here on a dendrite somewhere. The difference here is what kind of stimulus it is. Postsynaptic potentials, we're dealing with synapses between neurons. And we're talking about the cell that's at the sort of receiving end of the synapse, so we call it the postsynaptic cell. So a postsynaptic potential is a change in membrane potential on that cell at the end of a synapse. Whereas receptor potentials, these are basically what our sensory neurons do, and they're changing their membrane potential in response to receiving whatever stimulus they're kind of hooked up to receive. So let me show you an example. So here's where we have a chemical stimulus. This would be a receptor potential. In this case, so imagine this is going on in your nose, so your olfactory epithelium. You've got all kinds of little neurons in there that have very specific little channels, and some of these channels will bind to a chemical input. So in this case, this is chemical X. This could be like what they put in the Powerpuff Girls cocktail. Whatever this is some smell that's come into your nose and it's impacting on the olfactory epithelium. And it happens to bump into a receptor that is specifically designed to pick up that particular chemical. Maybe it's a sweet-smelling chemical or a sour-smelling chemical, or I don't know, some other kind of fragrance, some foul-smelling chemical, but it's binding to a receptor. And when it does so, in this case, what I'm showing, the way this thing is working, is that it's going to cause this channel to open. And in this case, this is a ligand-gated sodium channel. When this channel opens, that causes sodium ions to start rushing in. And if sodium ions start rushing in, they will then cause a local depolarization in the neuron's membrane. This would be a receptor potential, again, like odor reception, if we're talking about sensory neurons. Alternatively, if this thing, instead of having been some kind of chemical that was out in the air, if this had been a neurotransmitter binding here, we would have just created a postsynaptic potential. And some postsynaptic potentials work in this exact same way, more or less, where they bind to a receptor, the receptor opens a channel, and the result is a change in the membrane potential at that spot. Is that okay so far? So all we've done is we exposed a neuron to a stimulus that it's reactive to. It has bound that stimulus, or that stimulus has changed a receptor out on a dendrite, and we've changed the membrane potential in that little specific spot, a local potential way out on a dendrite. So this next picture this is my attempt at some depiction of this. So this is supposed to be the cell body of a neuron. This is one dendrite. I only drew one for sake of clarity. But this is supposed to be one dendrite sticking off of that cell body. And so what happened on the prior slide has happened in this one little spot right there. So there's some receptors there that bound their ligand or their whatever their stimulus is. Could be light, could be whatever. And as a result, they've opened and they've allowed a bunch of sodium ions to come in. And it's usually sodium ions coming in. That happened right there. And so what I'm showing you in this picture, this is a little electrode, and it's punched into the membrane of the dendrite at that point, and we're measuring the membrane potential change. And it's a pretty substantial inward flux of sodium ions, so we have shifted all the way up to about plus 75 millivolts in this area. It had been like down at like minus 60. So we've got a massive depolarization, but it happened right here. The thing that's kind of interesting, though, is that there's no real way to propagate this efficiently into the cell body. Some of these sodium ions that come in can spread down the dendrite. Some are going to go the wrong way, though. As they spread down, some of them kind of leak out. And so what ends up happening is what starts as a really strong disturbance over here, by the time it gets to the cell body, it's a pretty modest disturbance. So now, we started at minus 60. So now instead of going from minus 60 all the way to plus 75, we're going up from minus 60 to minus 30, which is still an upward deflection. That's actually still pretty good. But it's a pretty modest amount of stimulus that ever gets down into this area of the neuron. So I'm out of time, but the question for next week will be, how is it that these disturbances that barely even get to the cell body, how do those get ultimately result in a change in memory potential down on the cell body that's meaningful? All right, so I'm out of time. Sorry, again, I went a minute over.